All right, so I'm good. So today is my great pleasure to introduce our speaker, George Wells. George is assistant professor at Northwestern University, and I think it's fair to say that he's doing some of the most exciting work out there on nutrient removal in wastewater treatment systems. He's doing really cool things at the sort of larger pilot scale at the wastewater treatment plant. He's doing really neat molecular level work in the lab, and so he really covers the whole gamut. And I think he's doing some very cool stuff that he'll talk to us about today. He got his undergraduate degree from Rice University in chemical engineering and environmental engineering. And then he went to Stanford for his PhD and worked with Craig Criddle there and with the co-advisor Chris Francis, who's a microbiologist. He then did a postdoc at EAVAG in Switzerland for a couple of years and then came back over here. And he is actually from Fergus Falls, Minnesota. And so he had to get at least a little closer than California and Texas. So he came back partway to the Midwest anyway. So with that, I'll turn it over to George. Thank you.
Perfect. Well, thank you very much, Paige. Really a pleasure to be here. Thank you for the very gracious introduction. It is really nice to be back in Minnesota. I am a native Minnesotan. By the way, when I was in Switzerland, very few people knew where the Twin Cities or Fergus Falls were. And so I would start out, well, it's near Fargo. They didn't know. It's near Twin Cities. They didn't know that. It's near Chicago. So it is kind of near Chicago, I guess, right? So really a pleasure to be here. What I'd like to do today is to give you a bit of an introduction to some of the things that we're doing. Did I not turn on my mic? There are technical difficulties here. All right. See if that's working. All right, perfect. Okay, so what I'd like to do is to give you a little bit of an introduction to some of the things that we're doing at Northwestern, trying to address what I like to call the nitrogen challenge. In particular, what I want to do is to walk through a couple of new players in microbial nitrogen cycling, tell you about a couple of bioprocesses that we're looking into, and emphasize a new paradigm in the quest for sustainable nutrient pollution prevention. I think a number of the folks in this department are looking at this paradigm as well. So let me introduce this paradigm. Why do I have this word paradigm in my title? Let me introduce that to you first. What I'm really talking about is a paradigm shift in the way that we as a field and the way that we as a society view waste. Right? So this is a re-envisioning of what waste is, moving towards resource recovery. Much of what we do in my group is looking at wastewater, and so I want to focus in on what I mean by this resource recovery paradigm in wastewater. What do we do with wastewater? Well, at present, we take wastewater that's high in organic matter, high in nutrients, nitrogen, and phosphorus. In general, we send this to a centralized wastewater treatment plant. We expend quite a bit of energy, quite a bit of money to remove these so-called contaminants from water, and then, for the most part, we release this water to a receiving water body. This is a case of what Bruce Rittman has termed misplaced resources, or he actually calls them misplaced electrons. This organic matter, these nutrients, the water itself in wastewater, these are potential resources that we can think about recovering. And I think that's where we're going as a field, both in the research laboratory and increasingly in practice as well. And so what does this new paradigm actually look like? Let's take a very simplified view of what a rerouting of misplaced resources and closing of the engineered water cycle might look like. We have used water, which is just a semantic reframing of wastewater. This is actually what wastewater is called in Singapore, right? So it's not used it's not wastewater, it's not waste, it's just used. It's used water. And so we can think about recovering organic matter and nutrients. We can send this used water to an urban biorefinery where we extract useful resources. We can think about extracting energy, extracting nutrients, extracting materials, potentially producing platform chemicals, producing water. And where it makes sense, we can think about then reusing that water, so taking the clean water and reusing it, thereby closing the engineered water cycle. So this idea of resource recovery provides a pretty comprehensive framework for much of what we think about in my group. We're interested in both microbial ecology, which is the fundamental study of microbial community structure, microbial community dynamics, interactions, and emergent function in both impacted natural and engineered systems. And we're interested in the field of environmental biotechnology, where we ask, how can we put these microbial communities to work to do useful services for society? But really, most of the work that we do in my group is at this intersection between microbial ecology and environmental biotechnology, where we ask, how can we leverage new understanding of the microbial world to develop new strategies or new technologies for sustainable environmental and public health protection, coupled to this newer goal of resource recovery from waste. So this is basically a wrapper for everything we do in my group. Now, much of what we do in my group focuses on nutrients, and in particular nitrogen, which is the focus of the couple of stories I want to delve into today. And I want to tell you a little bit about why we're interested in nitrogen. What do I mean by this nitrogen challenge that I have in my title? To get at this question, I want to look at a plot on the x-axis of year, so the last 100 years or so. On the y-axis, we're looking at human population in billions. That's this blue line right here. We're also looking in this pink line at total anthropogenic reactive nitrogen input to the terrestrial environment. And what I want you to note is that over the last 100 years or so, both of these parameters have really ballooned. They've increased quite dramatically. And this basically exponential rise or the start of this exponential rise overlaps relatively well with the invention of a very important industrial process, the Haber-Bosch process for synthetic nitrogen fixation, for fertilizer production, right? So at present, human alterations have more than doubled the rate of input of nitrogen to the terrestrial environment, more than doubled the rate of input to a natural biogeochemical cycle. So as you might imagine, with a perturbation of this size, there are a number of negative unintended consequences. Why are we interested in addressing this problem of nitrogen pollution, of excess nitrogen flowing into the environment? First, ammonia displays toxicity to aquatic life. Any of you that have fish in an aquarium at home, you know that you need to monitor ammonia and actually total nitrogen as well in aquaria to protect fish. Ammonia also has a fairly high oxygen demand, about 4.57 milligrams of oxygen per milligram of nitrogen. There's a related problem of algal growth and eutrophication and resulting hypoxia in nitrogen-limited systems. Probably the most famous example of this, at least in North America, is the so-called dead zone, or the hypoxic zone that arises seasonally around the mouth of the Mississippi River in the Gulf of Mexico. There are emerging concerns about emissions of the potent greenhouse gas nitrous oxide, and I know there's a bit of work here at the University of Minnesota and Sebastian Barron's group looking at nitrous oxide. I'm going to come back to this molecule here, nitrous oxide, at the end of my talk. We're going to talk a little bit more about that. And there are also public health concerns, namely concerns about methemoglobinemia, which is caused by excess nitrite or nitrate in drinking water, and harmful algal blooms that cause release of cyanobacterial toxins. Here's a picture of harmful algal bloom in Lake Erie a couple of years ago. So I think it's clear that this excess, this doubling of the rate of nitrogen flowing into the terrestrial environment has a number of critical challenges that we as a field are now trying to address. In fact, there was a very nice publication that came out about 8 or 9 years ago from the National Academy of Engineering, highlighting 14 grand challenges facing the engineering community in the 21st century. And prominent among these challenges is management of the nitrogen cycle. So with that broad introduction to resource recovery and to the nitrogen challenge, to nitrogen pollution and why we're concerned about that, what I'd like to do is to step into 3 different stories, 3 different vignettes that I think will hopefully illustrate some of the activities that we're doing in my group, that we've done over the last 3.5 years or so. What I'd like to do is I'd like to start by telling you a story about population synchrony in nitrifying activated sludge bioreactors. This is a bit on the fundamental microbial ecology side, looking at ecological patterns of community dynamics in nitrifying activated sludge bioreactors. I'd then like to step into a much more applied project looking at Anammox bioprocesses for sustainable nitrogen removal. And I'll finish up the last couple of minutes of my talk talking about our efforts towards bioenergy production from waste nitrogen. This is a story that revolves again around this molecule, nitrous oxide. So let's begin then by looking at regional population synchrony and nitrifying activated sludge bioreactors. I want to make the link first to nitrogen cycling, microbial nitrogen cycling. And that link comes from this word here, nitrification. So let me give you just a little bit of background on what I mean by nitrogen cycling. What we're looking at on screen is our classical picture of the global biogeochemical nitrogen cycle. And when I say nitrogen cycle, I really mean microbial nitrogen cycle. It turns out that the vast majority of transformations of nitrogen in nature are driven by microbes. And so what I've done on screen is I've plotted a number of different nitrogenous compounds. We have ammonia, nitrites, nitrates, and nitrogen gas. And I've also indicated the oxidation states of nitrogen in all of these compounds. Now, our conventional or classical view of microbial nitrogen cycling tells us that turnover of reactive nitrogen, by reactive nitrogen I mean everything other than N2, nitrogen gas, turnover of reactive nitrogen in both natural and engineered systems is driven by two broad microbial processes. The first is called nitrification. This is an aerobic process, so it requires oxygen. And this in turn is driven by two groups of microbes, two groups of bacteria, the aerobic ammonia oxidizing bacteria and the aerobic nitrite oxidizing bacteria. These convert ammonia to nitrates for complete removal of reactive nitrogen from either natural or engineered systems. This conventional picture of nitrogen cycling tells us that we have to rely on denitrifying bacteria, process of denitrification, which is respiration of nitrate to nitrogen gas. And so it turns out that indeed as engineers, when we think about the two dominant point sources of nitrogen to the environment, that's municipal and industrial wastewater, we leverage these processes, nitrification and denitrification. So indeed, nitrifying activated sludge bioreactors are, I would argue, a cornerstone of our urban water quality control infrastructure. Activated sludge bioreactors are thought to be the largest example of biotechnology by volume in the world today. There are about 16,000 activated sludge bioreactors in the US alone, treating about 100 billion gallons of wastewater per day. These are relatively effective, actually quite effective technologies, but they are, or reactors, but they are also energy intensive. They account for about 3 to 4% of nationwide electricity use. Now, the purpose of activated sludge bioreactors is to intake low-quality water, elevate the quality of the water, that is, remove toxins, remove organic matter, remove nutrients, remove pathogens, such that the water is safe to release to a receiving water body or increasingly to reuse. On screen, we're looking at one of these 16,000 activated sludge bioreactors. This is actually in California. And as with all activated sludge bioreactors, the emergent function, the ecosystem function, or in engineering parlance, the process performance in this system is driven entirely by a highly complex, highly dynamic, but little understood community of microbes, of bacteria and archaea and microeukaryotes and probably viruses and phage as well. And over the last several years, much of my work has focused on trying to understand ecological forcings, community dynamics in activated sludge bioreactors, and then attempting to link that to emergent process performance. Specifically, one of the main classes of questions, let's say, that I'm interested in looking at is the relative influence of deterministic versus stochastic community assembly mechanisms. And we're getting a bit into the weeds of ecological theory here, so I want to tell you a little bit about what I mean here. Here's what I mean. In both natural and engineered microbial communities, community composition and dynamics have been proposed to be influenced by a number of different processes. These processes can be grouped, broadly speaking, into two overall processes. The first are deterministic, or niche-based processes, things like environmental factors, temperature, for example, or reactor operational conditions, dissolved oxygen, for example, or pH. Conversely, the second broad class of processes is stochastic, or neutral processes, things like random drift or immigration. The key point is that the relative importance of these two classes of community assembly mechanisms are little understood. And so this is the broad area that we're trying to address. The specific research question that I want to talk about today is a relatively simple one. What we're trying to ask is, do microbial communities in activated sludge bioreactors and regionally associated activated sludge bioreactors display synchrony in community dynamics and population dynamics? So if population 1, if taxa 1 goes up in one activated sludge bioreactor, if you go 5 miles down the road, does it go up in that reactor as well? And so this is a relatively simple question, but it hasn't, to our knowledge, been asked. And it's an important question, we think, because if we found evidence for regional population synchrony, this would be strong evidence for a dominant role of deterministic drivers of microbial community structure. So, relatively simple question. Do activated sludge microbial communities display regional population synchrony? How did we actually answer this? We looked at a map of the closest activated sludge bioreactors, nitrifying activated sludge bioreactors, around Evanston, which is the city in which Northwestern is. And we decided to sample these activated sludge bioreactors weekly. So we chose 6 of these, 6 full-scale nitrifying activated sludge bioreactors. We sampled them weekly for a year. We put the biomass in a freezer. Eventually, we extracted DNA, and we subjected these to what's called 16S rRNA amplicon sequencing. I'm going to skip over the methods a little bit just for the sake of time, because I want to tell you a bit about what we learned. But I'm happy to discuss the details of this after the talk. In the end, we ended up with about 22.5 million sequences, about 75,000 different DNA sequences per sample. And in total, I guess the first lesson that we learned, and this is not terribly surprising, but I do think it's interesting, is that activated sludge is an astonishingly diverse community of microorganisms. We detected about 19,000 or so different OTUs. OTUs is operational taxonomic units. This is microbial ecology speak for approximately species. We're not really sure how to define species, so we call them OTUs. So 19,000 or so different OTUs detected in at least one time point. So let's see if we can answer this question. Do we see regional population synchrony in activated sludge bioreactors? And I want to ask this at a number of different scales, 3 different scales actually. The first scale I want to ask this at is a very simple scale, and that is at the scale of alpha diversity. Alpha diversity, you can think of this as being approximately analogous to species richness, the number of species that we detect in any time point. What we're looking at on screen here is on the x-axis we have time of sampling, so a year of sampling. On the y-axis, we're looking at one of the metrics of alpha diversity that we looked at. We looked at a range. This is face phylogenetic diversity. And the colored lines, these dotted lines here, are results from the 6 different plants that we sampled from. What I want to point out, 2 key points. The first is that in all of the plants that we looked at, all of the nitrifying activated sludge bioreactors, we saw relatively high diversity in the fall, October and November. Going into winter, we saw a pretty steep decline in diversity, so December, January, February, March, and then a recovery and increase in diversity going back into spring and summer. So putative hints of seasonality in microbial community structure. I'll point out this is 1 year of sampling, and so we can't actually say for certain that it's seasonal. You just can't say that from 1 year, but there are hints of seasonality here. More interesting than this, though, I think, is that these dynamics in alpha diversity are strongly correlated between individual plants. So we see a very strong signal of synchrony of correlated shifts in alpha diversity between our the different plants that we looked at. So yes, indeed, at this level of alpha diversity, we see strong and statistically significant evidence for synchrony. So that's great. That's interesting, I think. But maybe a more interesting question is, do we see this pattern of synchrony repeated when we look at community structure or beta diversity? Do we see synchronized dynamics in community structure? And I'm going to put up a plot that probably many of you have seen similar plots to this before, but I still think it looks something like modern art. So I'm going to walk you through what we're looking at here. What we're looking at is a representation of community structure, right? A histogram representing community structure over time in our 6 activated sludge bioreactors. We have sampling time points on the x-axis. So week of sampling. On the y-axis, we're looking at the relative abundance of the 15 most abundant bacterial classes that we detected in our nitrifying activated sludge bioreactors. And what I've done is I've plotted the average across all 6 plants for these 15 bacterial classes, because even at this relatively coarse level of phylogenetic resolution, we see nearly the same abundance and nearly the same dynamics across the board for all of these classes in every reactor that we looked at. So very strong signal and statistically significant signal of synchrony. Also, the other thing I wanted to point out in this plot is that we also see a putative signal for seasonality in community structure. And so let me give you an example of that. If I can draw your eyes to the blue bar here, this is the beta proteobacterial class. This is the dominant class in every reactor and every time point that we looked at, but you'll note that there's some ups and downs here. So in all of the reactors that we looked at, we saw an increase in abundance of beta proteobacteria during the winter and a decline going into summer. Conversely, in about 11 of the 15 classes, we saw a statistically significant decrease in abundance in the winter and a corresponding increase going into summer. So again, we've seen statistically significant signals for synchrony at the alpha diversity level, at the beta diversity level. When we're just looking at this coarse level of resolution of bacterial classes, we can ask the same question at the species or OTU level at a much finer scale of resolution, and we get a very similar answer. What I'm showing on screen is a density plot of the distribution of pairwise correlation coefficients between distinct taxa in different plants. So that's in blue here. In green, we're looking at a bootstrap distribution of that same data set. So green is what we would expect to see if there was no signal of synchrony between these reactors. What you'll note is that we see a pretty significant rightward shift, right? So towards positive correlation coefficients in our actual data distribution. This is a statistically significant shift indicating relatively strong synchrony in populations at the OTU level between nitrifying activated sludge bioreactors. But there's a wrinkle in this story, relatively interesting wrinkle that we didn't quite expect. And the wrinkle is that what we're looking at on screen here are results from what we call the core microbiome. The core microbiome is the set of taxa that are present in every single time point in every single reactor that we looked at. And it turns out that this core microbiome is relatively low in diversity. So it encompasses only about 134 taxa, OTUs. Remember, we have 19,000 total that we detected in at least one time point. But these 134 OTUs represent the majority, just a small majority, but a little bit over 50% of the abundance of sequence reads. So the abundant, these are the highly abundant taxa. So strong signal of synchrony in these core OTUs. We can ask what happens in the rare OTUs as well, and we get an interesting answer. We do indeed find a statistically significant signal for synchrony when we look at these rare OTUs, but it's markedly weaker than for the core OTUs. So we appear to have abundance or frequency-dependent synchrony in community and population dynamics in activated sludge. So what does this mean? I want to give you a couple of takeaway points for this story. I think what we've shown so far, I think what we can conclude is that complex and highly diverse microbial communities and nitrifying activated sludge bioreactors, these key reactors controlling organic matter removal and nitrogen removal from wastewater, are highly diverse and display strong and significant patterns of regional population synchrony. We think this is important because it is strong evidence for a dominance of deterministic and therefore predictable drivers of community assembly. But there is this wrinkle. The wrinkle is that we see abundance-dependent community and population synchrony, and that suggests that stochastic processes may play an important role in structuring the rare biosphere in activated sludge. So with that, what I'd like to do is switch gears now and take off my ecology or my microbial ecology hat and put on my bioprocess engineering hat. And I want to tell you about a much more applied project that focuses on Anammox. So Anammox bioprocesses for sustainable nitrogen removal. So what do I mean by Anammox? I want to give you a little bit of background about Anammox. And to do that, we're going to go back to our picture of the classical microbial nitrogen cycle. And I want to make the point that one of the reasons it's been so exciting for me and so interesting to work in the realm of microbial nitrogen cycling over the last 10 or 15 years or so is that we've seen a number of unexpected and surprising discoveries that have really shaken our understanding of the key players in this nitrogen cycle. So I'm going to tell you about, or I guess I'm going to mention 3 discoveries over the last 10 or 15 years, or 20 years actually, that I think are particularly relevant to the work that we're doing. The first one I'm just going to mention very quickly, and that was the discovery about 10 years ago of an archaeal route for aerobic ammonia oxidation. This is very interesting because our conventional view of microbial nitrogen cycling tells us that a very small group of bacteria, this is a view going back about 100 years, very small group of bacteria are the only microbes, the only organisms on Earth that are capable of nitrification. So this is a discovery in a completely different domain of life of this metabolism, this capacity for ammonia oxidation. This was a major focus of my PhD work. It's still a focus of some of what we do in my group today, but I'm not going to go into detail about that in my talk. Happy to discuss this afterwards. The reason I'm not going to go into detail about this is I want to focus on a discovery that has maybe more immediate application in the engineering realm. And I want to point out that in this classical view of microbial nitrogen cycling, there are 2 key assumptions. The first assumption is that ammonia turnover, ammonia transformation, only occurs under aerobic conditions. In the absence of oxygen, in anoxic or anaerobic conditions, ammonia is inert, right? That's the first assumption. The second one is that for complete removal of reactive nitrogen, we have to rely, as engineers or in natural systems as well, on denitrifiers, nitrate reduction to nitrogen gas. Without denitrifiers, there's no conversion back to nitrogen. This assumption, these 2 assumptions were turned on their heads about 15, 20 years ago by the discovery of an anaerobic route for ammonia oxidation. First reported in 1995. This is a discovery that was greeted with great skepticism in the environmental microbiology community, but it was really nailed down as a microbial, novel microbial metabolism linked to so-called Anammox bacteria, anaerobic ammonia oxidizing bacteria, in a nice Nature paper in 1999. So what are these Anammox bacteria? What do they actually catalyze? They catalyze a shortcut in the nitrogen cycle. So they catalyze ammonia oxidation directly to nitrogen gas. Instead of linking ammonia oxidation to oxygen, they couple this oxidation to reduction of nitrites. Now, in the subsequent 15, 20 years, there's been quite a bit learned, very exciting information or lessons learned about the metabolism and the biochemistry and the ecology of Anammox in both natural and engineered systems, and this is certainly something that we're interested in. But I want to keep our focus on engineering. I want to keep our focus on how we actually apply Anammox bacteria in practice. And indeed, this is a question that engineers began asking very soon after the discovery of Anammox. And I want to show you why that is. To do that, let's take a look at our conventional routes for nitrogen removal from wastewater. Conventionally, we rely on these classical microbial processes of nitrification, ammonia oxidation to nitrates via this key intermediate, nitrite, NO2-. For complete removal, then, we rely on coupling nitrification to denitrification, the reduction of nitrate to nitrogen gas. And I'll point out 2 things in this simplified stoichiometry for nitrification. Denitrification, the first is nitrification requires quite a bit of oxygen. So 2 moles of oxygen per mole of ammonia. The second thing is that we require quite a bit of organic matter, generally, for denitrification. That's what I'm representing by COD here as well. What happens if we consider the possibility of adding Anammox into this process? What we can think about doing is we can think about oxidizing just a portion of the ammonia to nitrite. So not all the way to nitrate, just to nitrite. We call this process nitritation. We can then think about oxidizing the remainder of the ammonia, about 50%, directly to nitrogen gas, leveraging the activity of Anammox bacteria. And there are 2 key advantages of these so-called nitritation Anammox bioprocesses compared to conventional nitrogen removal systems. The first is that nitrotation Anammox processes decrease oxygen requirements by about 60%, or offer the possibility to decrease oxygen requirements by about 60%. And this is important because aeration can account for the majority of energy use in typical wastewater treatment plants. Anything we can do to reduce aeration requirements can pay huge dividends in terms of energy use. The second advantage, and I think this is a little bit less recognized, is that nitrotation Anammox processes effectively decouple removal of carbon and nitrogen from wastewater. What I mean by that is we've essentially cut out our requirement for organic matter, for COD here. And this is important because we can think about potentially recovering that organic matter, recovering that COD as a value-added product. We can think about biogas generation, that's sort of the low-hanging fruit, but moving forward we can think about producing value-added products, we can think about bioplastics production, platform chemical production, liquid biofuels, et cetera. So there's a clear link to resource recovery with nitrotation Anammox processes. So I think it's clear that nitrotation Anammox processes present great promise as a sustainable alternative to conventional nitrogen removal, but there are a number of key challenges that remain to be addressed before these are ready for prime time, especially for what's called mainstream wastewater treatment. I'm going to delve into the weeds just a little bit to explain what I mean by mainstream treatment, and then I'm going to tell you a little bit about what we've learned so far in our efforts towards addressing these challenges. So nitrotation Anammox processes are very new. They have been applied at full scale in quite a number of installations for treatment of high-strength, high-temperature waste streams. These are predominantly what's called anaerobic digester supernatants. If you look at the bottom of the screen here, we're looking at liquid that comes off of centrifuges after anaerobic digestion, high temperature and high ammonia concentrations. That's great. Nitrotation Anammox processes work relatively well in these so-called side stream treatment units. The problem is that the majority of the nitrogen load in a typical wastewater treatment plant is not in the side stream. It's in the mainstream. It's in the actual municipal wastewater. And so the holy grail, I would say, of Anammox Environmental Biotechnology's application to this mainstream wastewater, municipal wastewater, that is characterized by some pretty challenging conditions. We have low temperatures, or lower temperatures certainly, 10 to 20 degrees Celsius, low ammonia concentrations, low substrate concentrations, and in some cases, most cases, we have elevated organic to nitrogen ratios. So what are the main challenges to application of Anammox processes in the mainstream? I'm going to list 4 of these that we're trying to address. The first is maintenance of high levels of extremely slow-growing Anammox biomass and activity. There are a range of estimates for doubling times for Anammox, but the most commonly referenced minimum doubling time is 11 days. 11 days is a doubling time. So these are extremely slow-growing microbes. Retaining Anammox biomass and activity is a very key challenge. The second is maintenance of performance or stable ecosystem function under low temperature and dynamic conditions. The third, this is really the scourge of all what I call shortcut nitrogen removal processes. This is outcompetition or suppression of unwanted microbial populations, namely the nitrite oxidizing bacteria. So we want to outcompete nitrite oxidizing bacteria. And the final challenge that we're addressing, and I'm not going to go into this today, is we're trying to explore routes for coupling shortcuts or Anammox-based nitrogen removal to biological phosphorus removal and recovery. Very interesting. I'm really excited about this challenge, but I'm not going to talk about it in great detail today. The approach that we've taken to trying to address these challenges is we've partnered with the Metropolitan Water Reclamation District of Greater Chicago to pilot test, bench pilot scale test, 2 bioprocess variations for mainstream nitrotation Anammox, again combined with biological phosphorus removal, but I'm going to focus on nitrogen removal here. Here's a picture of our setup. It's a bit complicated here, but we have a couple of different nitrotation Anammox reactors. We have an upstream COD and phosphorus removal reactor. I'm going to focus on the nitrotation Anammox reactors here. A key point I want to make is that we are operating these reactors on site at a local water reclamation plant. And so because of that, we're able to use actual wastewater, primary effluent. And so we see actual realistic conditions, dynamics and composition, temperature, et cetera, that are seen in practice. So what are these two bioprocess variations that we're looking at? How do they differ? The key difference is the type of microbial aggregate that we're selecting for in these systems. The first of these processes is a biofilm reactor. It's actually a hybrid reactor, but it's predominantly biofilm. So I'm just going to refer to it as a biofilm reactor from here on out, in which we select for growth of microbial biomass on quite a number of small plastic biofilm carriers. So a biofilm reactor. We're comparing this directly against a parallel suspended growth reactor in which most of the biomass is floccular, although we do have a minor fraction, about 5% or maybe a little bit less, of the biomass that forms really interesting granules in this system. So we have biofilm versus suspended growth. We're asking, can we maintain activity of Anammox, and can we maintain at least some of the performance characteristics that we desire, routing nitrogen through nitrotation Anammox, the nitrotation Anammox pathway. So the first question I want to ask is, can we actually maintain Anammox activity over an extended time period under mainstream conditions? And to answer that question, let's take a look at dates on the x-axis. We've been operating these systems for about 8 months or so. I'll mention that this is an ongoing project. So we are, I would say, at the end of phase 1 of this project. So I'm going to show you a couple of results, lessons learned from what we've done so far, and then give you a bit of information about some ongoing work and where we think this project is going. So data on the x-axis, we have nitrogen depletion rates driven by Anammox during Anammox activity assays on the y-axis here. In green, we're looking at results for the biofilm system. In purple, we're looking at the suspended growth system. Let's start with the biofilm system in green. What we see, in both of these systems were inoculated with biomass from a side stream system. So we have plenty of Anammox in the inoculum. In the biofilm system, we see a moderate decline in activity of Anammox over the first couple of months of operation, but then a pretty decent recovery of activity over the last 5 months or so. So what it looks like for our biofilm system is that indeed we are able to maintain robust Anammox activity under mainstream conditions for an extended time period. Conversely, let's take a look at our suspended growth process. We see fairly good activity to begin with, but then a dramatic decline in activity and no real recovery of that activity over time. I will point out that technically we have maintained Anammox activity in this reactor. We have measurable about 50 milligrams nitrogen per liter per day, but it's dramatically lower than in the inoculum, in the suspended growth process. So based on activity measurements, yes, we can maintain Anammox activity under mainstream conditions, but the biofilm reactor is the clear winner. We can also ask this question in terms of performance. Are we able to select for at least some of the performance characteristics that we desire under mainstream conditions? Are we routing some of the nitrogen transformation through nitrotation Anammox pathway, the nitrotation Anammox pathway? And so let's take a look first at results for our biofilm reactor. We have data on the x-axis again. On the y-axis, we're looking at TIN concentration. This is total inorganic nitrogen. Influent is in blue, and red we have effluent. Because we have an upstream COD removal reactor, TIN removal, total inorganic nitrogen removal, is a relatively good proxy for nitrogen removal via nitrotation Anammox pathway. So what do we see? On average, we see ammonia removal of about 75% and total inorganic nitrogen removal around 50%. So the key takeaway here is, yes, we're seeing a substantial fraction of nitrogen transformation converted over a long time period through the nitrotation Anammox pathway. It's certainly not where we want this to be in the end, but it's headed in the right direction. So I would call this proof of feasibility of maintenance of at least some of the performance characteristics that we want under mainstream conditions. Conversely, in our suspended growth reactor, we see much poorer performance over time. Not surprising given the decrease in Anammox activity. Ammonia removal is about 75%, but we're seeing much of this converted directly to nitrates. We're seeing about 33% TIN removal. So key takeaway from this process level examination is we've shown retention of Anammox activity over an extended time period under mainstream conditions, and retention of Anammox, nitrotation Anammox performance, at least a substantial fraction of nitrogen transformation through nitrotation Anammox in the biofilm reactor. So I would say we're at the sort of proof of feasibility stage. Much poorer performance in the suspended growth reactor. I want to spend a couple of minutes telling you where we're going now. Really what we're trying to do is we're trying to link these patterns of macro scale process performance to mesoscale variation in aggregate architecture and microscale variation in spatial distribution of microbial populations. Let me just show you a bit of results from this. What we're looking at is one of these nitrotation Anammox biofilm carriers from a side stream system. Remember, side stream means high temperature, high substrate conditions, ideal conditions for nitrotation Anammox. What we've done is we zero in on one of these void spaces for biofilm growth. We cryosection it. So we're looking at just one of these voids. We cryosection and we use fluorescence in situ hybridization with fluorescently labeled oligonucleotide probes to look at spatial variation, spatial distribution of microbial populations. In red, we see Anammox. And we see that we have a very thick, robust layer of Anammox that's overlain by a thin layer of this green population here. This is our desired aerobic ammonia oxidizing bacteria. And in blue, we have NOB. NOB are nitrite oxidizing bacteria. These are undesired. We don't want these in this system. And indeed, you see that we have very few blue spots here, right? So this is what a very healthy nitrotation Anammox biofilm looks like. What do we see in our mainstream biofilm system, our bench or pilot scale system? What we see is still a very healthy, robust layer of Anammox, very thick layer of Anammox. We see quite a number of aerobic ammonia oxidizing bacteria, but we see an accumulation of this blue population, the nitrite oxidizing bacteria, the unwanted nitrite oxidizing bacteria. And so the key message here is that our strategies that we're using to try to suppress NOB, to try to select for nitrotation Anammox bioreactor performance is partially successful, right? We're seeing some nitrogen flow through that pathway, but it's not quite enough. We still have a nitrite oxidizing bacterial an NOB problem. This is even more pronounced for our suspended growth system, where we're seeing in the inoculum very little NOB. After a couple of months of operation, we still see these very dense aggregates that are dominated, we call them granules, that are dominated by Anammox, but we see an accumulation in the surrounding loose floccular phase of nitrite oxidizing bacteria. So where we're at this point is I think we've shown feasibility of Anammox activity and nitrotation Anammox performance, at least a substantial fraction of nitrogen transformation through nitrotation Anammox metabolism in the biofilm system. So we're moving from feasibility to optimization, really focusing in on this scourge of shortcut nitrogen removal processes, which is NOB suppression. And so we're looking at a couple of different control and operating strategies in progress now, trying to more aggressively outcompete NOB. So aeration control and modulation of aggregate architecture are the two levers that we're trying to turn. Happy to go into more detail about these operational strategies after the talk. For the sake of time, though, I want to switch gears. And I think I have time to get through my last short vignette here. And I want to stay on a relatively applied track here, but I want to get a little bit more blue sky. And I want to recognize that, yes, nitrotation Anammox processes are very promising. They present a route to saving quite a bit of energy for nitrogen removal, and they're linked to resource recovery because we free up carbon for resource recovery, but we're not explicitly trying to recover value from the nitrogen itself. We're not trying to recover the nitrogen as a value-added product. And so that's really the driver behind this last story that I'm going to tell you about, where we're looking at bioenergy recovery from waste nitrogen. The story revolves around this molecule here, nitrous oxide. Nitrous oxide is a natural component of the atmosphere. Its pre-industrial concentration in the atmosphere was about 270 ppb. Over the last 200 years or so, this has increased to about 330 ppb. This is a problem or at least it was a problem before the election, because N2O has, it's a very potent greenhouse gas. It has a global warming potential about 310 times that of carbon dioxide, and accounts for about 8% of worldwide greenhouse gas emissions. There are a number of significant sources of anthropogenic nitrous oxide emissions, including wastewater management. Where does nitrous oxide actually come from in wastewater management? It comes from a couple of different microbial metabolisms, including denitrification. So we're switching to a different part of this microbial nitrogen cycle. Let's take a look at a simplified stoichiometry of denitrification. We have nitrate reduction to nitrogen gas via a number of intermediates, including nitrites, nitric oxide, and here we are, nitrous oxide. And it turns out that under some conditions, this last step in denitrification is inhibited or even completely absent from the denitrification pathway. And so because of the fact that nitrous oxide is a potent greenhouse gas, there has been quite a bit of research in the last 10 years or so looking at ways to minimize emissions of nitrous oxide from both engineered and natural environments. And indeed, this is an area of interest for me as well. But a couple of years ago, with a collaborator, we realized that nitrous oxide is actually a pretty potent source of energy as well. It's used as a monopropellant in rocket engines. It's also used in nitrous injection engines, so race cars. I gave a presentation to a student group several months ago, and I asked them, where have you heard of nitrous oxide before? And one of the students raised their hand and said, Fast and the Furious. So nitrous injection engines, right? So nitrous oxide is actually a pretty powerful bioenergy molecule. So with this in mind, with our collaborators, we've come up with an integrated approach to trying to leverage this energy potential in nitrous oxide. We call this the coupled aerobic anoxic nitrous decomposition operation. It's quite the mouthful, but it comes up with a nice acronym, the CAN-DO process. We like to be very positive in our bioprocess names. The basic idea behind the CAN-DO process is that we're trying to promote removal of nitrogen, but then conversion, maximization of conversion to nitrous oxide. We capture the nitrous oxide in our overall schematic and use that as a bioenergy source. The strategy that we've settled on over the last couple of years in denitrifying systems is to employ what's called a feast-famine regime, where we alternate between feeding high organic matter and, or high BOD and high nitrite feed streams. And so we alternate high and low organic matter in these systems. Here's a picture of the reactor that we're operating at Northwestern. This has been operating for about 2 years or so. And if we zoom in, this is a TEM image. What we can see is that we have a diverse community that we selected for, but we select for a community that's capable of accumulating these white dots here. These are polyhydroxyalkanoate inclusions, or intercellular storage polymers, bacterial fat, essentially. So the key research question that we're asking at present is, can we take this CAN-DO process for nitrogen removal and bioenergy recovery and combine it with phosphorus removal via selection for what are called denitrifying polyphosphate accumulating organisms? So I'm mixing up the waters a little bit more and adding in another, not newly discovered, but little understood class of nitrogen cycling microorganisms. I want to introduce you to these organisms just for a minute. And then I'll show you a couple of results and wrap up. Polyphosphate accumulating organisms are the key functional guild in what are called enhanced biological phosphorus removal processes, processes for removal and increasingly recovery of phosphorus from water. Most polyphosphate accumulating organisms, or PAUs, affiliate with so-called Accumulobacter phosphatus. Some of these PAUs are capable of denitrification. We call these DPAUs, denitrifying polyphosphate accumulating organisms, but this class of PAUs is relatively little understood. We're interested in DPAUs because they offer a critical opportunity to couple nitrogen removal to phosphorus removal and recovery. So let's orient ourselves in our picture of the changing nitrogen cycle. Where are we? We're in the denitrification branch here. So denitrifying polyphosphate accumulating organisms. So what have we learned? We have this lab scale reactor. We've operated it for close to 2 years now. What have we seen? The first thing I'll point out, or the first question I'll ask, is have we actually been able to enrich for these Accumulobacter phosphatus, these PAUs? We're looking at on screen here at day of operation. We're just looking at a half year of operation here. On the y-axis, we're looking at relative abundance of POWs. These are Acinetobacter phosphatus, monitored by fish, and we see an increase from about 5% up to 70 or even higher percent, 70 to 80% POWs. So indeed, we are able to enrich for POWs in this reactor. That's great. But do we see the activity that we want? Do we see denitrifying polyphosphate accumulation? I'm going to show you the results from a representative cycle in this reactor. We're looking at time on the x-axis. The y-axis, we're looking at a number of products and reactants. It's a bit of a messy plot, so I want to just draw your attention to the orange line here. This is phosphate. For POW activity, what we expect is release of phosphorus under anaerobic conditions and uptake under aerobic, or for denitrifying POWs, uptake under anoxic conditions. So indeed, we are seeing activity of POWs. We see release under anaerobic conditions and uptake, at least partial uptake, under anoxic conditions. So we're seeing the activity that we want. The key question is this associated with high levels of nitrous oxide production? Do we see production of nitrous oxide that we can potentially recover as a bioenergy molecule? The short answer is yes. We're looking at 3 representative cycles here, and I'm just showing you nitrite, which is the influent nitrogen source. Nitrite is reduced, and the majority of that nitrite is reduced just to nitrous oxide, this purple bar here. This is reproducible and stable over time. At present, we're seeing about 70 to 80% conversion to nitrous oxide as a novel bioenergy source. So again, I want to emphasize that this is a proof of concept, blue sky approach to capturing value from nitrogen. I don't expect this process to be in place at full scale at your local wastewater treatment plants next year. We have some work to do still on this, but I'm excited about it because I think it's an interesting example of trying to rethink what is a waste and how do we recover value from that waste. For the sake of time, a couple minutes over where I want it to be. Sake of time, I'm going to back up and try to give you a couple of takeaway points from these 3 vignettes that I've gone through today. Hopefully I've convinced you that newly discovered microbial players are dramatically changing our understanding of the global biogeochemical nitrogen cycle. And as engineers, we have an opportunity to take advantage, to leverage this new understanding of microbial nitrogen cycling to develop new and sustainable bioprocesses for both nitrogen pollution prevention and for this new goal of resource recovery from waste. Or recovery of misplaced resources. I'll wrap up with an acknowledgement slide. I owe a thank you to many, many different people, and I don't have time to go through all of them. I just want to point out a couple. I need to update this picture, actually. This is a picture of my group, but it's expanded a bit. Han Gao is the PhD student who did the work on all of the can-do work that I showed today. Alex Rosenthal is a postdoc in my group. Paul Roots is a PhD student. They did the work, are still doing the work on the nitrotation Anammox application to the mainstream. And Jim Griffin is responsible for the regional population synchrony story that I told you about. So thank you very much, and I would welcome any questions from you.
And please wait till I get to you with the microphone.
Okay. This is outside my field, and I read abstracts, not papers. Okay? But there's quite a bit of research on non-biological removal of phosphorus going on.
Yep.
Are they consistent with both your Anammox concept and your can-do, or are they actually, I don't mean economically competing, I'm talking about could they be done in series or in parallel, et cetera?
So great question. We are actually just starting a line of research funded by WRF to ask that question for recovery of phosphorus from chemical precipitation with ferric chloride, ferrous chloride, for example. Certainly, chemical precipitation is very common in the field at this point. It is directly compatible, should be directly compatible with Anammox-based processes, because if we separate carbon and phosphorus removal from nitrogen removal, we just need a mechanism of removing phosphorus upstream of the Anammox process. For the can-do process, if we're not targeting phosphorus recovery, I would think that it would be compatible. If we're talking about phosphorus recovery, it's much easier to recover phosphorus at this point if we use biological phosphorus removal than chemical phosphorus removal. And there's some other sustainability arguments for biological phosphorus removal as well. Very good question, though.
In your Anammox section, you talked about possible techniques on how to use it, specifically the differing amounts of oxygen over time. Could you explain that maybe a little more?
Yeah. So the approach that we've been using to date for NOB out competition is continuous low dissolved oxygen and elevated ammonia residual. So we still have some ammonia in our effluent, which is something we have to address at some point, but at this point we're just trying to select for Anammox, right? The continuous low dissolved oxygen is, as we've seen, partially successful in the biofilm system, not so successful in the suspended growth system. And so we're transitioning to what's called transient anoxia, which is intermittent high aeration. And there's some evidence based on pure culture studies that NOB nitrate oxidizing bacteria are less adapted to basically turning off and turning on their metabolic machinery than ammonia oxidizing bacteria. And so this is yet to be really robustly shown in practice, I think, in a mixed microbial community, but that's the lever we're trying to turn with that. Yeah.
You are observing 75% removal of ammonia in the process of Anammox nitration. Do you know how much is being actually removed by Anammox and how much is removed by the other?
Yeah, so that's a really good, so ammonia removal or total inorganic nitrogen removal, which you're interested in ammonia? So ammonia actually is relatively easy to fractionate because we also have nitrate measurements. And so we know the total rate of nitrification and we know the total inorganic nitrogen removal, and so we can just subtract them basically. The more difficult removal, and so it's about 50% for the biofilm reactor and about 75% full nitrification in the suspended growth reactor. So much better in the biofilm reactor. The more difficult processes to distinguish are Anammox from denitrification. We were talking about isotope-based methods last night. That's a great way to distinguish them. It's very difficult in practice, and so the way that we do this is we look at total inorganic nitrogen removal, we look at COD removal, and COD is removed both by aerobic heterotrophs and probably a minor fraction by denitrifiers in this reactor. As a conservative estimate, we say all of it is removed by denitrifiers, so the minimum amount of nitrogen removed by Anammox is this amount. Does that make sense? And it turns out that the maximum possible nitrogen removal by denitrifiers, based on COD measurements in both of our reactors, is about 20% of the total inorganic nitrogen removal, 15 to 20%. So a large majority is Anammox.
All right, this better be our last question, I think.
I have about 800, but I'll just ask one, I guess. So this is in regards to your population dynamics experiments. You said that you wanted to tie population structure to function of the plants. Was that kind of like a secondary goal? Because there wasn't really any...
Yeah. So really good question. So I mentioned that as a very broad goal of why we're interested in looking at microbial ecology in activated sludge. Some of my previous work has tried to tie nitrification failure to shifts in community structure. This particular project that I talked about, we didn't really delve into that aspect that much. We do have some data, this is in the paper that just came out, looking at links between community dynamics and sludge bulking, so settling in activated sludge. And so I would say that is the most direct connection to function in this project.
Okay, so you didn't compare like whether or not your core OTUs that you detected in each plant were more important for function than the rare OTUs?
Oh, good question. We didn't assess the functional significance of the rare versus core OTUs. It's a really good question, but no, we did not assess that. Be something great to follow up on, though.
All right. Well, we're about to be inundated with biomechanics people, so we should go ahead and thank George.